blood, right? Give you guys a few moments if you're following along in your slides to find this. Okay, so this is an important process because if you guys get any sort of cut and start bleeding, your body hopefully should naturally be able to stop it. So for example, let's say you get like a small finger cut, like a paper cut. How long do you think it takes for you to stop bleeding from like a small paper cut? Yeah, maybe just a few minutes, right? So this could be potentially a very fast process. Of course, sometimes our body can't handle it on its own and we need help for the clotting. But let's go through the process in which our body is able to form a nice scab and clot your blood. All right, do you guys remember which type of blood cells help us clot blood?
Platelets.
Yeah, the platelets, right? We have red blood cells, white blood cells, and platelets. So platelets are our main blood cells that are going to help us clot the blood. So here's what's going to happen. So just to give you guys a picture here, this is up here your blood vessel on the top, and then this is where you see a break in the wall of the blood vessel, right? So that's where you have your small cut. So what's the first thing that's going to happen is that these little platelets, which look like little stars that are in your blood, are going to come and gather at the site where you got the cut. So that's the first step. We say the platelets form a plug. So they basically come together and try to plug up the hole in the blood vessel wall. But the problem is the platelets themselves are not strong enough to patch up that hole. So what they have to do is bring in a protein called fibrin. So in this picture, fibrin is the green protein, and they're long and narrow. And what the fibrin does is it weaves through the platelets, and so it forms a nicer, stronger plug. So it's the platelet plus the fibrin that's needed to get a nice, strong plug in your blood vessel wall to stop the bleeding. Right? So platelets alone can't do it. All right, so one of our major questions then is how do we get that fibrin to form and come to the cut? The platelets are just there, right? The platelets are in your blood, so they just naturally go. But the process of making fibrin is a little more complicated. You just don't naturally have fibrin in your blood. So we're going to take a look, and I'm going to show you a few of the key steps involved in making that fibrin protein. Remember, no fibrin protein, your platelets aren't going to be strong enough to form the clot. All right, there's two things that initiate the making of fibrin, and it really depends on how you got your cut. So, two ways you can get a cut, extrinsically or intrinsically. So what does extrinsic mean? Outside, right? So think X and no outside. So if you get a cut from the outside, imagine that's like I'm here and I get a paper cut, right? Or I'm cooking dinner and I cut myself with a knife. Right? If you get the cut from the outside, you physically see the cut from the outside, that is going to initiate the extrinsic mechanism. And your tissue is damaged. Your tissue from the surface of your skin is damaged, right? You cut through it. And then you start bleeding. Intrinsic mechanism. That is if there's no tissue damage from the outside. In other words, from the inside, your blood vessel is broken and you start bleeding from the inside. Anyone know common ways in which you can get an intrinsic bleeding starting?
Blunt trauma.
So aneurysm might be one, like if you have a stroke going to your brain and your blood vessel bursts. How else might you get an internal bleeding?
Blunt trauma.
What's
Like a blow that doesn't break the skin.
Yep, a blow, like think about a car accident, right? If you get in a bad car accident, you have a strong blow to your body, you might not physically see any bleeding on the outside, but they might still take you into the hospital and do some scans because they're worried about internal bleeding, right? So any strong trauma or blows to the body, you could possibly cut your blood vessels from the inside, but externally you wouldn't see it. Another great example would be a bruise. Right? Have you guys ever gotten a bruise, but yet your skin wasn't broken? You're like, how did I get that? Right? When you have a bruise, let's say you bumped yourself and you didn't know, again, your blood vessel broke from the inside, but yet there was no outside external tissue damage. So that's extrinsic versus intrinsic. But no matter which way you got your cut in the blood vessel, both of them have to initiate the formation of fibrin. So let's see how that works. Okay, so what you have is when you get the blood vessel cut, it activates this protein right here called the prothrombin activator. The reason why this protein is important is actually an enzyme. And what it does is it takes this protein that you naturally have in your blood. So everyone has this prothrombin protein floating around in their blood plasma. When you get cut, you activate the prothrombin activator. This enzyme will convert your natural prothrombin into another protein called thrombin. Here's why thrombin is important. Thrombin serves as an enzyme, and what it does is it takes this other protein that you naturally have in your body, in the blood plasma, called fibrinogen. And it'll convert fibrinogen into the fibrin protein that you need to then form your clot. All right? So again, in your blood plasma, you naturally have prothrombin and fibrinogen. They're just floating around, ready to be used. To activate this whole process, you have to get your cut. Your cut makes this enzyme active, prothrombin activator. The enzyme prothrombin activator converts prothrombin into the protein thrombin. Thrombin serves as an enzyme that will convert your fibrinogen into the final protein fibrin, and the fibrin is what weaves through your platelets to make the final clot that you need. And again, this process can happen really quickly, right? With a quick paper cut, you'll notice results in minutes, right? 3 to 6 minutes, you've already got this nice clot forming, and hopefully you're stopping the bleeding, right? And eventually that turns into a scab. Right, so questions on that?
You said prothrombin and fibrinogen are naturally in our bloodstream.
Yeah, exactly. Both of these, prothrombin and fibrinogen, you naturally have in your blood plasma. They just need to be activated. So I've given you kind of the short, simple pathway. Think of it like a domino effect. But in reality, if you look in the book and look at the whole process, there's actually 30 plus factors involved in this whole pathway. So I've given you just kind of the few key ones, but there's actually 30 different types of proteins that need to all be activated in this domino effect to ultimately get fibrin. And your book actually gives you this table where it's giving you some of the main factors involved. So I've only pointed out a few of the main ones. But notice there's all these different enzymes involved in making this fibrin protein, which in this picture is all the brown weaving through. So why do you think there's so many clotting factors? Why do you need 30 plus clotting factors? Yeah.
Maybe because sometimes you could be hurt like maybe a lot more, like sometimes a certain cut might need just a little bit of those, but like if you're in a car accident, you might need more.
Yeah, that's actually a great observation. Maybe if your cut is really big or larger, you might need more factors involved to get a stronger cut. Yeah, that's a really good guess. Any other reason?
Maybe depending on where your cut is in the body, because there's so many different tissues.
Okay, good. Maybe depending where your cut is, you'll activate different factors.
Maybe you want to be careful about clotting because even though it's good, you don't want to just have clots floating around.
Okay, and so finally, maybe you want to be careful about clotting, right? You don't want to prematurely form a clot because you don't want clots floating around your blood. It can stop your circulation. So those are all great ideas. Let me add one more thing. No matter where the cut is, no matter how little or how big the cut is, you still actually have to go through all 30 of these factors. Yeah. So the real reason, the actual reason why is what Rachel actually suggested, is that you have 30 factors because you want to go through each one of these steps very carefully and double check to make sure you absolutely want a blood clot. Right? In no circumstance would you want to form a premature blood clot, because a premature blood clot, if that gets in your blood vessel, that's actually going to stop your blood circulation, right? And that's bad. So you want to make sure with every step of the way, you're like, do I really need this clot? Yes. Do I really need it? Yes. You go through like 30 checkpoints, and then finally you can get fibrin and make your final clot. Right? So again, it's to prevent premature clotting because that would be really dangerous. And sometimes people are more susceptible to clots in their blood. You might have heard of something called a thrombus or an embolus, where they actually get these floating blood clots which can stop their blood circulation, again, which is not good. Right? So that's why we need so many of them. Yeah, Bethlehem.
So are we going to use like all 30 clotting factors at the same time?
Yeah, great question. So Bethlehem said, are we going to use all 30 factors at the same time? Yes. Not at precisely the same time, a domino effect, so one after the other. But in order to get any clot, yes, you will need to use all 30 factors. Any clot, anywhere, no matter how small or how big. Yeah, Oscar.
So what happens to the people that have hemophilia?
Yeah, fantastic question. What happens to the people who have hemophilia? So does anyone know what hemophilia is?
Bleeding disorder?
It's when they can't form blood clots properly. So a problem with hemophiliacs or a risk is they get a small cut and they just keep bleeding and bleeding and bleeding. So they can't go through. Be a little closer. They can't go through this process and they can't get the fibrin. So the problem with hemophiliacs actually is right here. I said we all naturally have fibrinogen in our blood plasma, ready to be converted into fibrin for the clot. Hemophiliacs lack this really critical protein in their blood. So if they don't have fibrinogen, can they form fibrin? No. If they can't form fibrin, can they form a proper clot? No, which means they'll keep bleeding even at the smallest cut. But one great thing is that because we know hemophiliacs are lacking fibrinogen, what can we give them to help?
Fibrinogen.
Yeah, you can just give them that. And actually it's a really popular biomanufacturing business right up here nearby, like Grifols is one of the major companies. What they do is they take blood plasma donations. So you guys can go and just donate your blood plasma, not even your blood cells. They filter out the blood plasma, and what they do is they take your blood plasma and filter it out for your fibrinogen. They bottle it up, and then they can give that to medication, the hemophiliacs. So it's actually something that they have a good understanding of, and they actually can help fix that. Great question, Oscar. All right. Okay. So let's say you have the opposite problem. So hemophiliacs means you can't clot your blood naturally. Let's say you have the opposite problem and you're susceptible to premature blood clots. Right? You're more likely to form a blood clot. So there's ways to actually stop this process. Right? Let's say you don't want a blood clot. We're going to do what we call dissolution of coagulation. Coagulation is another way of saying your blood is coagulating or clotting or clumping together. Dissolution means to get rid of. So this is to get rid of a blood clot. All right. So to get rid of a blood clot, we have to somehow stop this pathway. Right? So one thing we can do is actually a common thing is to use what we call anticoagulants. So based on their name, what do you think they're going to do? Yeah, they're going to stop the clot, right? Anti is against, coagulation is the clot. So these are just enzymes that inhibit or stop the clot. One very common one that's administered in a lot of clinics, you might have heard of, it's called heparin. So heparin, again, inhibits blood clots, and how it does so is it specifically stops thrombin. So if we stop thrombin, will we make it to fibrin? No, and no fibrin, right? No clot. So again, this is for people who are susceptible to prematurely forming clots, and you don't want that again. You don't want excess clots in your blood. Or if they find that someone has a thrombus or a clot floating around in their blood, they can administer heparin. And another kind of way of describing heparin is a blood thinner. Right? Again, thinning out the blood so there's no clots in it. Okay. All right. So that wraps up hemostasis. Remember, hemostasis is the process of forming a blood clot. You need platelet plugs with fibrin to form the clot. And you know now that hemostasis can occur extrinsically or intrinsically and needs many factors to work. blood clotting. Okay. So with that in mind, let's move on to our next lecture, which is understanding the heart. Remember, cardiovascular system is blood, heart, and blood vessels. So the second lecture we have is on the heart, and then our last lecture will be on the blood vessels, where we'll deal more with things like blood pressure. I'm gonna start passing around the roll sheet. All right, so let's take a look here.